Hi, I'm Ben Haller, and today I'm going to talk about SLiM, which is a framework for scriptable, interactive, fast evolutionary modeling. And I'll introduce SLiM, and then I'll focus briefly on a couple of new features that we're excited about. Sounds like the mic is kicking in a little better now. So does that work? No. How do I do the next slide? There we go. All right. So, SLiM is a team effort. Philip Messer and I are the primary developers of SLiM, but my co-authors here all contributed particularly to the tree sequence recording feature that I'm going to discuss in a bit. And Peter Ralph has also been instrumental to the development of support for continuous space models in SLiM, which I'll also mention. So SLiM is often described as software for forward genetic simulation. And so what is meant by that exactly? Forward means that the simulation runs forward in time from an initial ancestral state, and that's in contrast to the coalescent, for example, which runs backward in time. Genetic means that SLiM simulates explicit genetic loci at specific positions along the chromosome, and that's in contrast to more phenotypic simulations that lack that kind of explicit genetics. And simulation here means that SLiM runs individual-based models in which the birth, life, and death of explicit individuals is simulated, as opposed to analytical models that often work at a higher level, such as population means, or variances, allele frequencies, or overall rates of change. So there are lots of reasons to use simulations. If you have empirical population genomic data, then simulations might allow you to analyze the past evolutionary forces that produce the patterns that you see in your empirical data set, and even to predict the future evolution in your system. If you're a theorist, simulations can let you predict the consequences of a theory or compare multiple theories to see which produces the best fit to the phenomena that you're trying to explain. And simulations can also be useful for those that are working on statistical methods as a way to test the accuracy, bias, or power of a statistical method using simulated data that's generated by known evolutionary processes, or as a way to generate training and test data sets for machine learning. But there are various competing methods of modeling and simulation, such as analytical models and coalescent simulations. So why do forward simulations in particular? The answer is essentially that evolution is complex. If all of the simplifying assumptions of analytical or coalescent models apply to your study system, then that's great. But real-world biology typically involves all sorts of complexity, such as the examples listed here that violate those assumptions to some degree. So if you want to model a system that contains this kind of complexity, then forward simulation is often necessary because it allows you to model those specific aspects of your system that make it interesting and different. So if I've sold you on forward simulation as a worthwhile tool, you might still ask why use SLiM in particular rather than some other software package, or perhaps even writing your own model from scratch. There are several reasons. One reason is that SLiM provides an interactive graphical modeling environment called SLiM GUI. SLiM GUI provides live visualization of your model's dynamics as it runs. It also has a full-featured code editor, online documentation at your fingertips, and much more. So the value of this for model development, for debugging, and for open-ended exploratory work is really enormous. SLiM GUI makes it really easy for new users to get started with SLiM too. A second reason is that SLiM is very flexible and customizable because it's scriptable. Your model is written in a scripting language called ADOS, allowing you to do almost anything you want at different points during the generation cycle and to override SLiM's default behaviors with custom script. ADOS is easy to learn with very standard syntax similar to R, as you can see here. And a typical SLiM model can be expressed in a page or less of ADOS code, so it's much simpler than trying to write a model yourself in a language like C++. SLiM is doing most of the work for you under the hood. A third reason is that SLiM is very fast. I've put months and months of work into optimizing SLiM's performance, and if you use SLiM, you benefit from that work automatically. If you use SLiM's tree sequence recording feature, which I'll describe in a few slides, the speed benefits are so large that for large models, SLiM with tree sequence recording, as shown by the green curve here, has a runtime that's actually as fast as the coalescent, shown here in red. And since the coalescent is sort of the gold standard for speed, that is no small thing. So that means that you no longer have to make the difficult choice between the speed of the coalescent versus the biological realism of forward simulation. With tree sequence recording in SLiM, you can have your cake and eat it too. A final reason to use SLiM is that it's open source on GitHub under the GPL license. It's free to use, and if you wish to do so, you can even get involved in its development. So I'm going to spend very little time on this slide, but here's an example SLiM script for a simple neutral model, just so that you can see what all of this looks like in practice. The top block initializes the simulation's genetic structure and mutation rate and recombination rate. The next block starts the simulation by adding a new subpopulation, and then the last three blocks at the bottom just produce output in specific generations using some standard output methods. So this model is quite trivial, obviously, but with just a few more lines of script, you can take it in a million different directions because of the flexibility of scripting. And the manual for SLiM has over 100 different examples of such models. Okay, so that's it for the introduction. I want to spend the rest of my time talking about 4 new-ish features in SLiM that we're very excited about. Support for continuous space in SLiM was added in version 2.3. In version 3.0, we added support for non-Wright Fisher models and tree sequence recording. And very recently, in version 3.3, we added support for nucleotide-based models. And I'll talk briefly about each of these. So in continuous space models, individuals shown here as the little kind of colored blobs or squares, they live on a continuous landscape rather than in discrete subpopulations. The landscape can have any of a variety of boundary conditions, and SLiM provides a sophisticated spatial interaction engine to make spatial interactions like competition and spatial mate choice really easy and fast. Under the hood, the interaction engine uses KD trees to find nearby neighbors of individuals and sparse arrays to store information about the interactions in a memory-efficient way. But you don't need to worry about those implementation details. You just work with the high-level scripting interface of the interaction engine, and you get the speed benefits for free. You can also define landscape maps, such as the discrete valued map shown at the lower right that divides the world into water and land, or the continuous valued map at upper right that here defines variation in the local environmental variable. You can define multiple landscape maps, and those maps can influence your model's dynamics in any way that you wish, such as by affecting the movement or the survival of individuals. Okay, the second new feature is non-Wright Fisher models. So the Wright Fisher model was the foundation of all SLiM models prior to version 3. Now, however, you can make a non-Wright Fisher model or non-WF model in SLiM instead, which provides much more flexibility and realism. So in non-WF models, population size is not fixed as it is in Wright Fisher models, but is instead emergent from the births and deaths that are occurring in the model. So it's much more biologically realistic in that sense. Population regulation is not automatic as it is in Wright Fisher models, but instead is scripted. So a non-WF model might simulate forces like competition, predation, or migration that together regulate the population size. In Wright Fisher models, fitness is expressed through the probability of mating, but in non-Wright Fisher models, fitness by default affects survival, which is usually more biologically realistic. You can also model mating success in your script if you want to. And in non-Wright Fisher models, generations can overlap, and age structure is emergent from the births and deaths that occur in your model. Finally, migration is scripted in non-Wright Fisher models, so your model script can move an individual from one subpopulation to another at any point in time, as compared to Wright Fisher models where migration only occurs for newly created juveniles. So together, all of this basically means that non-Wright Fisher models are more individual-based, more emergent, and more biologically realistic in general. And so the ability to create non-Wright Fisher models in SLiM greatly increases SLiM's flexibility and generality. Okay, tree sequence recording is the third new feature and probably the most complicated aspect of SLiM. In essence, it keeps track of the full ancestry tree at every position along the chromosome, and that's accessible to you later in Python. So an example tree sequence is shown here at the bottom. There are 5 extant individuals at the bottom of each of those trees at the end of the simulation run that's shown here. And the ancestry along the chromosome from left to right shows that there are 4 different histories because of the recombination events that caused genetic material from different ancestors to be combined together into the same genome. The ancestry information that's recorded by tree sequence recording is all available after the simulation is completed using the Python packages MS-Prime, TSKit, and PySlim. So that's useful in itself. Having that ancestry information can be tremendously useful for analysis, but it's also very useful for performance reasons. It means that neutral mutations can actually be added across the tree sequence after forward simulation has completed, and that saves enormous amounts of computation during the forward simulation. It means that a neutral burn-in period can often be simulated with the coalescent in MS-Prime and then used as a starting point for your non-neutral simulation in SLiM. And it means that a non-neutral SLiM simulation can actually be done with no burn-in at all, and then a neutral burn-in can be added with the coalescent after the fact using a method that we call recapitation. So all of that is a little bit complex, but suffice to say that these methods can speed up the overall simulation by several orders of magnitude, and that can bring even very large forward simulations within reach. So finally, in SLiM 3.3, which was released just over a month ago, we've added support for nucleotide-based models. In such models, SLiM tracks the nucleotide sequence of every genome, and mutations have an associated nucleotide, so they're really SNPs. Mutation rates here are sequence dependent. You can use standard mutational models like Jukes Cantor or Kimura 1980, or you can define your own mutational model. The mutational model can even depend upon the trinucleotide context surrounding the mutating position, so it's easy to simulate things like an elevated mutation rate at CPG sites. And a fairly realistic model of gene conversion is provided, and that includes heteroduplex mismatch repair within complex gene conversion tracts, as well as support for GC-biased gene conversion. So this should really open the door to all sorts of new possibilities in SLiM. Okay, so there's over 100 recipes in the manual. I've barely scratched the surface of SLiM's features and capabilities. This slide basically shows some pretty plots and screenshots from a few of those recipes that are in the manual. There are resources for those who want to explore SLiM. The MesoLab website has reference sheets and a double-click installer for macOS. There's a discussion list where you can email me directly, and we've got 3 papers published this year, one on SLiM 3, one on tree sequence recording, and one that provides an introduction to SLiM 3 for beginners. There are workshops coming up, one in Umeå University in Sweden and one at the University of East Anglia in the UK. Both of those are still open for registration. There are 5-day workshops. In late-breaking news, there's going to also be a SLiM workshop at the City University of New York in November, and the registration for that is open now. Just go to the SLiM homepage and find the section on workshops and follow the instructions there. So I'd like to end by thanking the funding agencies that supported this work and the very long list of people who have contributed to SLiM in one way or another, and thank you for patiently listening to my rambling. I'd be happy to take questions, and anybody who wants a SLiM sticker for their laptop can get one from me afterwards, since this is the last session in the talks. So.
Do you have online course somewhere?
There's no online course as such, but the manual itself kind of serves as an online course to some extent, because it has so many example recipes. It starts off pretty simple and gets more complex. So I recommend working through the manual.